Yeah, I think it has started now, right?
Yes, I can see that.
Yeah, it started now. Okay, so we'll just begin.
Live streaming is there?
Yeah, we can
Yeah.
see it
Now.
on the screen.
Yeah.
Yes. Okay.
Now it's there.
Yeah. Okay. Thanks a lot, Dr. Pete. A very good afternoon to all present here. I'm Dr. Madhur Kulkarni. I'm currently working as a secretary of Executive Committee, Control Release Society's Indian chapter. On behalf of the Executive Committee, I extend warm welcome to you all for today's event. Before we begin the session, a few requests to all the attendees. Kindly mute your mics and please switch off your videos throughout the session. If you have any query or concern, please put it in the chat box and we will try to address it. Thanks a lot. The outline of today's session is that first we will be having a technical talk that will start now and it will be there till 4 p.m. The talk would be followed by the question answer session up to 4:15 p.m. And then onwards, we will proceed with the annual general meeting of CRS-IC. The topic for today's technical talk is oral delivery of biologics. And the invited speaker for it is Dr. Yogeshwar Bachau. The speaker's profile and abstract of his presentation has already been circulated to all, but I would still like to give a brief introduction of Dr. Yogesh. Dr. Yogeshwar Bachau is a pharmacist by training. After completing his PhD in pharmaceutics from the Institute of Chemical Technology, Mumbai, he worked in DACH region of Europe for around 15 years with different startup and mid-sized biotech and pharmaceutical companies. During this tenure, he has successfully managed pharmaceutical development of several projects from POC to phase 1, 2, and 3 clinical trials. He has a strong network of experts and CDMOs in Europe, USA, and Asia. In addition, he has 16 international publications, 8 patents, and 30 conference proceedings to his credit. Recently, he has edited a book for Wiley VCH Germany on innovative dosage form, design, and development at early stage. Dr. Bachau has been invited as a speaker for many international symposia around the globe. He is a full-time consultant with a German biotech company, AI Curis Anti-Infective Cures, since 2014. There he is responsible to deal with pharmaceutical development of new drugs in anti-infective segment. In addition, Yogesh has founded a pharmaceutical R&D consultancy firm, edX Pharma, in 2016. The prime objective of edX is to resolve complex issues in the pharmaceutical development of new drugs and life cycle management of approved drugs. He also supports a few clients in India to resolve the CMC issues. Dr. Bachau is a member of Executive Committee of Control Release Society's Indian chapter. With this introduction, I hand over the session to Dr. Yogesh.
Good afternoon, all. Thank you, Mother, for a very kind introduction. I think it's my pleasure to be here today to talk on this topic, oral delivery of biologics. Control Release Society Indian chapter. This is a platform I am associated with right from 2006, where I was in fact volunteer to organize the annual meetings. And since the association continues, so as I said, I'm also part of the executive committee. So I think the pleasure is double today if I have the possibility to discuss a little bit about this topic. So with your permission, I will start my talk today. Can you see the screen? And am I audible?
Yeah.
Yes.
Good. Okay. So let me deal with this. Okay. So the contents of today's talk will discuss briefly the market scenario of oral biologics. Then What are the limitations and challenges we face with respect to the biologics? What are the formulation considerations the formulation scientists should consider? What are the reported strategies or recent advances where we stand as of today? Then we'll also discuss little bit the clinical trial scenario, which are the, let's say, the technologies in the pipeline, where we expect some compounds or at least the technologies, the market in the very near future. And then we will try to conclude this session. So regarding the market scenario, as you can see on this slide, in the year 2018, we had the biologics market around 800 US million dollars, and it's expected to grow by a factor of 10 when we go 2026. So the growth rate is quite high. The reason for that, more and more molecules under clinical trials, under development, they are coming from the biologic segment. So as if now, if you see the overall scenario, we have more than 300 monoclonal antibodies, around 250 vaccines, and more than 100 other biologics, including the cells and gene therapies under clinical development. So what are the major drivers for this growth? There are more and more increasing partnerships and collaborations here. To give an example, in 2017, Rani Therapeutics had a deal with Shire to develop active transport technologies for factor 8A for the treatment of hemophilia. I mean, this is just one example. There are several collaborations. There are several technologies being explored, as we'll discuss during today's talk. And that's the major driver why the market still tries to increase. If you see the overall scenario, more than 50% of, let's say, the global the revenues are, as if today, from the pharmaceutical markets, they are coming from the biologics market. And there we certainly need to think then what could be the innovative dosage forms and technologies from the patient compliance point of view, from the cost effectiveness, because we all know for specific reasons, mainly the, let's say, the stability point of view, the protein's conformational structure we'll see later. As if today, most of the formulations we use for the proteins or biologics, they are the parenteral formulations, either to be administered through the IV route or maybe intramuscular route or subcutaneous route. And in terms of patient compliance, maybe it's highly desired that one should come with oral formulations. So that will have significant impact on the compliance as well as cost. But of course, it's not that easy. So this is something then really an interesting field from the pharmaceutical development point. And there's a lot of scope then to come up with the new options, how we can translate these parenteral formulations into the oral dosage form. So typical examples of these partnerships and collaborations, maybe we can think of this technology, SOMA, self-orienting multimeter scale applicator. The details of this technology I'll present later, but just in a nutshell, I can say this is a oral insulin drug delivery technology for type 1 diabetes. This was developed by Bob Langer at MIT, and they had a deal with Novo Nordisk. And the technology is moving quite well, and they are all set to enter into the clinic, and the concept is quite interesting. So I think if we have any innovative concept, and if we try to deliver this biologics by active transport technique, because passive transport is certainly not an easy deal, but then there is a lot of scope, and this is like a platform technology. If it works for a given compound, we can also try to explore for other biologics. So these are the blockbuster drugs, and as you can see, most of these, they are from the biologic segments, the monoclonal antibodies, different proteins. So that also indicates why it's very important to discuss today the biologics market and what we can bring as a value addition in terms of the formulation design. So on this slide, we see the forecast about the drugs which will achieve the blockbuster stage, and that is meant by the sales of over $1 billion by 2023. So most of the drugs, if you see, again, they are the monoclonal antibodies or proteins, and from the innovator companies like Roche, Novartis, or maybe AbbVie. And important to note here, this biologics, they are very specific in terms of the pharmacological response compared to the small molecules. So the adverse events are comparatively less compared to the small molecules. But at the same time, there's a lot of challenge in terms of delivery. It takes a lot of efforts and budget to come up with the stable formulation. So this has to be also kept in mind. And then we should also think what is the patent scenario, because let's see if we have to explore any oral technology for any approved protein, then one has to also think whether this drug is off patent or this is still patented. As you see, more and more drugs are going to be off patent, so that gives ample of opportunities for the generic companies even to think they can come with some biosimilars, or maybe they can explore this off patent proteins to come up with alternative process forms, maybe into the form of 5,5B2 scenario, where they can translate the parental formulation into the oral formulation. But the challenge is really to obtain the similar pharmacological response in terms of the PK/PD profile. So when we think of delivering the biologics like peptides, proteins, maybe vaccines by the oral route, what are the physiological barriers we are talking here? So the first thing we should try to consider the acidic environment and the degrading enzymes. We know when the any drug substance is administered through the oral route, it is reaching to the stomach, which has acidic pH between 1.2 to 3, depending on the disease state, whether it's a geriatric population, adult population, or maybe pediatric population, the pH will always vary. But at that particular acidic pH, the proteins are not very stable. So this is one thing to be considered. At that acidic pH, we may expect the protein molecules to undergo degradation. And once they are degraded, of course, we cannot expect the physiological effect there. Other important point to consider, the degrading enzymes. So in case of stomach, you do have the enzymes like pepsin, which may lead to the degradation of protein molecules. And when we think of lumen, the intestinal membrane, in intestine also a lot of peptidase enzymes are secreted into the epithelial layer of the intestine. Again, there are several proteolytic enzymes present. This is another barrier which leads to the degradation of the protein molecules. So this is to be kept in mind. Sorry. So, so this is something to be kept in mind when we are trying to develop the peptide or protein formulation. Then we do have the layer of intestinal fluid or the intestinal cell wall, and one of the layer is also mucosal layer. So mucosa is a mixture of the mucin, proteins, and ions. It has a thickness of 500 nanometers. So the surface of mucosal layer is quite lipophilic, and it is attached to the intestinal wall by the glycoprotein link. And this also presents quite significant barrier for the protein molecules. So this mucosal layer barrier should also be considered. So can you put on mute, please? So one of the strategy we need to consider. So maybe combination of mucolytic agents may be necessary, or when we are trying to design these dosage forms, we need to think really which particular portion of the GI tract we have to target for the delivery of biologics. And another important factor here, then when it crosses across the intestinal cell wall, the entire length of GI tract, where it is getting absorbed, because the maximum surface areas available, let's say the lower part of the intestine, where we do have the presence of villi and microvilli, but the distance between this microvilli is let's say around 25 nanometer, and then that doesn't really give sufficient space for the biologics to cross. Maybe the small molecules is a bit easier to cross through the surface, but for biologics it's not the case. And like small molecules, for biologics also it's very important that their absorption gets varied along the length of the GI tract. So when we are trying to design any formulation, it's very important we should know at which specific location of the GI tract we want drug to get absorbed, and based on that then one has to design the formulation. And another important aspect we have to consider, although we try to take care of this peptide or protein degradation here, we try to address the presence of mucosal layer, but the degradation of the protein is also possible when it is crossing the epithelial layer, because there is a presence of proteolytic enzymes in these layers. So the combination strategy is needed where we can think of using some enzyme inhibitors so that the protein drugs are not getting degraded during their passage to the epithelial cell layers. And once we are able to take care of all these barriers, maybe then we have the success that we are able to deliver the proteins or the biologics across the intestinal cells, and finally it reaches to the, let's say, the systemic circulation. So what are the typical formulation considers we should try to discuss when we are trying to discuss the formulations? So the proteins are quite delicate structure. These are the folded structure is most stable if you see in terms of the physiological activity from the structural point of view. So we should try to consider that when we are trying to develop this formulation, it will be in combination with excipient, it will be subject to stress conditions, and the chances are quite high this folded structure will not remain intact. The unfolding will take place. The moment the unfolding takes place, then there are some intermolecular or intramolecular bond formation, which may lead to some physical and chemical routes of degradation listed here, like association might happen. Maybe proteins will undergo denaturation, aggregation, depending on the stress factor we are using, depending on the nature of excipients, you may have some chemical degradation in the form of maybe hydrolysis, disulfide exchange, or oxidation. So this is something to be kept in mind when you are trying to design the formulations of protein molecules. So during the pre-formulation, it's very important to understand under which conditions, maybe pH, maybe nature of excipients, the temperature, our proteins is more stable. And once you have that knowledge, then I think there's a good background to come up with the formulation strategy in order to develop the stable protein formulation. And this is also driven whether you want to come up with the liquid formulation or the lyophilized formulation. Again, it becomes more complex because here we are discussing about oral delivery. When you want to think of developing a parenteral formulation, in that case liquid formulation is still okay because it can be kept at 2 to 8 degrees Celsius, maybe minus 20 degrees Celsius for a long time. It can be then diluted and kept in infusion bag prior to the administration. But when it's given by oral route, either it will be in the form of, I mean, majority of this active technology, if you see, they are in the form of capsule shell. So naturally, we can't keep the liquid formulation into the capsule. So at the end of the day, we need to convert that formulation to the solid state, maybe by using the lyophilization technology. And when we are trying to develop the lyophilized formulation, we will try to use some bulking agents, cryoprotectants. And during that stage, we have to make sure that this folded structure or 3D structure of protein is not disturbed and it should remain intact, and then only we can expect the desired physiological response. So all these aspects to be considered. So here I tried to list which are the physical parameters we should try to consider while developing the formulation. The solubility, the hydrophobicity of the drug is very important because that will really drive which kind of vehicle we want to use, what kind of excipients we need to keep the drug into the stable form. We also want to get rid of any self-association or aggregation. The molecular weight of the drug is very, very important because that will also decide. Imagine if you're dealing with any octapeptide or decapeptide, which is a small molecular weight compound. So compared to the proteins where you may have 25 to 30 kilodalton protein. So maybe peptides are getting absorbed better across the intestinal cell wall compared to your proteins. And if you are dealing with the vaccines, so anyway, these are quite bigger particles. So all this information is very critical to decide which typical therapeutic, which typical delivery technology we are going to use. And then it comes about the biological properties, because unlike the small molecules, the proteins, they do not really cross the cells to exert the physiological effect. They are mainly binding to the surface receptors. So they're really remaining on the cell surface. So any structural change will lead to that binding to the ligand. And you may not observe the desired physiological response. So this is something to be kept in mind. Then it comes to PK profiles, as we're discussing, majority of existing formulation, they are given by parenteral route. In this case, you are trying to shift from parental route to the oral route. So at the end of the day, you would like to have the more or less similar PK profile, then only desired pharmacodynamic response can be obtained. So this is something to be kept in mind. So to summarize, the formulation development of proteins or biologicals is very challenging due to their complex nature. The structure is quite sensitive towards these stress factors. So by optimizing the formulation with respect to the composition, so we can take care of these challenges. Sorry. So when we try to consider the oral administration of the proteins or biologicals, what are the typical pathways we're expecting for the permeation so that systemic circulation is possible? So the most obvious work one could expect is a transcellular passive transport where molecule is just passing across the cell wall, but due to its large molecular weight, due to its complex structure, it's not possible. Then we may rely on the paracellular passive transport, but this is also not very easily possible for the biologics. Maybe small molecules can pass through this paracellular passive transport, but these are linked to the tight junction proteins which are the membrane proteins translocated across the cells. And then only by achieving the active transport, we need to have some strategy so that these tight junctions are opening. Maybe we have some penetration enhancer so that these paracellular channels can be open, and then only it's possible to deliver the compounds across this paracellular route. Transcellular active transport is also another possibility, but in that case, then we have to use the penetration enhancers so that this tight epithelial layers on the surface of intestinal we can achieve the desired permeability so that the passive, the transcellular active transport is possible. So basically, the strategies we can think to improve the oral delivery of biologics are summarized on this slide. So the first option could be we try to protect these drugs from the acidic atmosphere across the GI tract and to avoid the enzymatic degradation. So the details we'll discuss in the subsequent slides. We can also think of changing the barrier properties of mucosa. So I said before, maybe the presence of mucolytic agents is very useful there. Then increasing the residence time of these biologics across the epithelial layers, typically the small intestines, is very important because that's where the maximum absorption of the drugs takes place. So this is something can be doable. Other option, then, as I was talking in the previous slide, we do have the tight junctions located between two cells which are like a paracellular route. So if we try to target that tight junctions by using penetration enhancers, that could be another possibility. We can increase the delivery of biologics. And here we try to see what happens when we are trying to deliver the drug across the oral route, typically for biologics. So when it is passing from the oral cavity to the stomach, this passage is quite faster, so it's very shorter transit time. So of course the absorption is very minimal during this time frame. When it's stomach, the pH is in the range of 1 to 4, so the transit time is also less. For the intestinal surface, we do have the pH in the range of 6.5 to 7. But again, for small intestine, it offers you quite high surface area, but the presence of digestive enzymes is quite high. The mucosal layer thickness is another barrier there. The fluid composition is quite complex. And in case of colon, you do have very low surface area, but the transit time is quite high there. The presence of digestive enzymes is also quite less. So for like small molecules, for the proteins also it's applicable. We cannot really generalize for all the proteins. Maybe the desired penetrations or permeation will be the same. So depending on the physical chemical properties of the particular drug, you need to see then which is the target site we are going to use. Maybe it could be stomach, it could be large intestine or small intestine, and depending on that, the suitable technology can be decided, or it could be selected for further trials. So on the right hand side of the slide, we can think, depending on the target site of delivery, we can think of the combination approaches, and it is more or less the combination of permeation enhancers, proteolytic enzyme inhibitors, the acid stable coating, In case of intestinal, we can think of mucolytics. Then, of course, we can also use the targeted nanoparticles or maybe liposomes or micro spares, depending on the drug loading, depending on the site of administration or the site of absorption. Sorry. And here, then I try to specify really what are the possible options when we are coming with the suitable dosage forms to deliver the biologics through the oral route. So we can think of co-administration of the protease inhibitors. Typically, these are the enzymes which lead to the degradation of the biologics, and absorption enhancers. So absorption enhancers are basically the permeation enhancers. And this combination can really help to decrease the degradation of proteins. And then once proteins are degraded, then it leads to improved permeability. Because imagine if you are not using permeation enhancers, only using protease inhibitors, okay, you will take care of the degradation of the protein. Protein will remain intact on the intestinal cell surface, maybe at the, around stomach cell wall. But if there are no absorption enhancers, naturally, then we are not getting rid of the barriers we are facing there. So this combination is very important there to use. Another option, maybe we can think of changing the physicochemical properties of these macromolecules, so that it's possible to improve their permeability. So we try to improve their stability against these enzymes. This approach may be easy to think, but in the practical terms, maybe not that easy. Maybe for small molecules, easy to achieve the structural changes. In case of proteins, it looks very complex. Another option we can think of using the advanced drug delivery systems such as nanoparticles, microparticles, or liposomes, so that we try to load these compounds within these vesicles. These vesicles then by taken by active transport route, or maybe this possibility this paracellular tight junction route can be used to deliver the drugs across the intestinal cell wall. In this case, the advantage is double-fold or twofold, because by encapsulating these molecules within this vesicle, within these particles, we try to take care of the degradation by the proteases. And as said before, the permeation could be easy when these molecules are coated within these delivery systems. The use of mucoadhesive polymers is very important because they try to increase the residence time of the biologics in contact with the intestinal surface, and that's how we have the possibility to increase the absorption. Then the site-specific delivery to the colon is very important, because as said before, in colon the number of protease enzymes are low. The pH is quite on the higher side, which is in favor of the protein molecule, and that could be very helpful to achieve the desired absorption. And then pro-drug approach, where we try to have the chemical linker to the drug molecule, so that we try to achieve the increased stability, increased permeability. But again, this approach also looks quite complex, because it's not so easy to achieve the pro-drug of protein molecules. So let's start to discuss with the co-administration of protease inhibitors, how we can increase the absorption of the proteins. So basically, in this case, we are using chemical substances, which are the inhibitors of the proteases enzymes we discussed so far. These proteases could be present in stomach as well as intestine. So what happens, the enzyme inhibitors are administered in combination with these biologics, and upon arrival into the GI tract, they try to bind to the enzymes, and once this enzyme inhibitor complex is formed, the activity of enzyme is inhibitor, that leads to the absorption of the intact protein across the epithelial cells. And that's how we try to achieve the systemic absorption. And in this case, it's very important which proteases we are going to inhibit. We don't want to inhibit the proteases, which are also important food digestion. So this specific interaction is very, very important. And the presence of proteases along the entire length of GI tract could not be same. So this is also important, at which location we are trying to achieve the delivery. It could vary with respect to the disease state the patient is. So this is also to be considered. And there are different selection criteria. So the specificity towards target enzyme should be considered, whether we are able to achieve to reach that target, where we want to achieve the absorption. This is also to be kept in mind. And at the end of the day, the drug is linked to the biologics, so that enzyme inhibitor should also have possibility to getting absorbed across the epithelial cells. So this is something to be kept in mind. Then another strategy is the co-administration of permission enhancers. So basically they try to act by different mechanisms. So some of the penetration enhancers, they are the chelators basically. So they just try to bind to the calcium present on the cell surfaces. So once the cells are deprived of calcium, they become more permeable, and that's how it's possible to increase the delivery of the proteins across the cell walls. The citric acid or maybe sodium editate could be best example of this strategy. Some of the surfactants or the bile salts, they act by changing the permeability of the cell surface, so cells becomes more permeable. Some of the penetration answers, they try to bind to the tight junctions. Once they bind to the tight junction, that leads to the opening of tight junctions, and then the delivery of the biologics is possible across this tight junction. So these are more or less 2 to 3 mechanisms we can use the permeation enhancers to increase the delivery of the proteins or biologics across the cells, and that's how we can increase the systemic circulation of this proteins or peptide. So in this case, again, it's very important. We don't want any non-selective permeability changes of the cells. Otherwise, we could have problems with the patient suffering from the celiac disease or irritable bowel syndrome. We definitely don't want to see any adverse events from this permeation enhancer. So the selection should be done quite carefully, because we also want to see the compatibility of the peptides, proteins under consideration with these penetration enhancers. And so as discussed before, there are a lot of regional variation for the absorption of proteins and peptides. So this is to be kept in mind, which particular location we are trying to deliver the proteins, whether we do have the tight junctions there, whether we are trying to target the tight epithelial cell layer junctions there. So this is also an aspect to be considered. And last but not least, the nature of delivery vehicles, whether we are trying to use the nanoparticles or liposomes, whether we are trying to use any active transport technologies. So this is also to be kept in mind, because it will also have some impact for the selection of permission enhancers. So from the literature, you can see phenylpiperazine compounds like SLS, they act quite very well as a permission enhancers. There are a lot of reports for the enhancement of insulin penetration by use of these permission enhancers. And the very interesting example I could find from the literature is of semaglutide, which is a GLP-1 receptor agonist. So this drugs is very highly effective to lower the glycated hemoglobin. It also facilitates the weight loss. And currently it is available as subcutaneous injection. Naturally, then it has some impact on the patient compliance, and that's the reason for its underutilization. So the researchers, they tried to develop the oral peptide formulation. In that case, they tried to use the excipient like sodium N hydroxybenzyl aminocaprylate. So it is basically the sort of capric acid they are using, so which is a penetration enhancer basically. So how it works? So this is given in the form of the tablet. So once the tablet is reaching to the stomach, this SNAC, the excipient they are using, it tries to have some buffering effect. By the virtue of that, it tries to change the pH in the microenvironment that leads to the inhibition of the activity of the proteolytic enzymes present there. And once that inhibition of enzymes is achieved, the drug is more or less non-covalently bound to the drug. So drug in combination with this excipient tries to traverse across the stomach cells, because it also has the penetration enhancing properties, and by virtue of that, the semaglutide is delivered across the stomach cell walls and it reaches to the systemic circulation. And upon once it reaches to systemic circulation, the drug is slowly dissociated from this SNAC, and that's how then it is available at the site of action. So it is a first oral formulation of this GLP-1 RA to be approved in the US. So very interesting technology. So on this slide, I tried to show the comparative data of the oral semaglutide against subcutaneous injection. You see, by using the oral route, they were able to achieve the similar Cmax values. Because the AUC, the area under curve, or the total observed physiological absorption for subcutaneous routes was comparatively high. But when you see the PD data, at least compared to oral route and subcutaneous route, they were able to get the equivalent response we will discuss in the next slide. So on the left-hand side of this slide, you can see they have done the studies at least in 8 different settings, so which is listed as a pioneer here. So basically, they tried to compare the pharmacodynamic response of semaglutide at different doses compared to placebo. They also tried to compare its response against the comparator drugs which act through the different mechanism, and they tried to see what is the overall impact on the lowering of this glycated hemoglobin as well as weight loss, which we discuss on the next slide. Sorry. So on the top, you can see the response in the form of the reduction in the glycated hemoglobin from the baseline when this oral semaglutide was compared with respect to the comparators. So as you can see, when semaglutide is compared with respect to the placebo, there's a significant reduction into this glycated hemoglobin. Same is the case with the different comparators they have checked. The oral semaglutide was behaving very nicely, so naturally, the desired absorption is quite, the obtained absorption is quite good through the oral route. They also tried to compare against the subcutaneous route. When you try to see the response compared in the form of the reduction in the body weight, the oral form of semaglutide is working quite well compared to the placebo group, compared to the different comparators they have used. We can conclude that during this program, the oral semaglutide is shown to be very effective for the patients to achieve the glycemic control and to reduce the body weight. And as the study was also conducted in the patient with the cardiovascular risk or cardiovascular diseases, in those patients it showed the safety profile very well acceptable, and there was a significant reduction in the cardiovascular death. Then what are the options if we think of modifying the physicochemical nature of the macromolecules, so that because the overall aim is really to increase their stability in contact with the proteolytic enzymes, and on the other side we are trying to improve their permeability across the intercell wall or maybe into the stomach. So the one of the options or one of the alternatives they have tried is pegylation. So PEG, we know it's a GRAS excipients, very well acceptable by FDA because its safety profile is quite good. It's soluble in water and organic solvents, so it is part of many oral or parenteral formulation. So once the pegylation of the protein leads to what improved solubility, the half-life extension is possible, the toxicity can be reduced in this case, immunogenicity risk is can be also reduced, and as discussed before, when the protein molecule is exposed to the high temperature, the pegylation really helps to achieve the robustness against those harsh conditions. It also provides protections against the proteolytic enzymes without impacting its biological activity. And if you see the literature, they have tried to use both branch chain as well as linear chain pegs for the pegylation, and the studies are done with the model compound as insulin. The results showed that branch pegs working quite well in terms of the desired stability, the improved permeability compared to the linear chain pegs. Another option is we can think of replacing the L amino acids by the D amino acids. So basically, we are just trying to use the different stereoisomer of typical amino acids, because once this stereochemistry is changed for that particular site of degradation, so maybe the given labile portion of that molecule is not available for the enzymatic degradation, and this can also significantly help to achieve the desired stability. One of the example is for LHRH hormone. They have tried to replace the L amino acid with the D amino acid at 6 to 10 position, at 6 or 10 position or both positions. The obtained protein was quite stable. To give an example, the non-change protein was just stable at 2 hours at 37 degrees Celsius. But when they tried to change this L amino acid with the D amino acid, the resulting protein was stable at 50 degrees Celsius for around 20 hours. So these are the effects you can really imagine when you are trying to change this typical stereochemistry at the site of degradation. Another option is to use hydrophobization. So in that case, we just try to achieve the desired hydrophobicity. One of the approach used there is to coat the polymers with the biodiesel polymers, like maybe carbopods or chitosan. This is another example. Sometimes they also try to use more lipophilic amino acid in the background of structures. The coating of this molecule, the coating of the molecules is also another option to achieve this hydrophobization. And overall aim is really to have these hydrophobic characters to improve their stability. And it also helps for the, let's say, the transcellular transport of these drugs across the cell layers. Sorry. Then we can also think of using the particular drug delivery systems to achieve the oral delivery of the biologics. And on this slide, I tried to list down the different examples. They are already into the clinic, some they are already into the market. So cyclosporine is one of the example, which is, let's say, the cyclic peptide. It has molecular weight 1,200. If you see the commercial formulation, it's micromerchant based formulations, neurals and immune, the typical composition I will discuss in the subsequent slide. So this is certainly possible if you have any possibility to load these molecules in the form of these lipophilic vehicles. We certainly have the possibility to improve the oral absorption. Then SLNs are also reported for insulin. You do have application of liposomes for insulin or growth hormone-like compounds. Then nanoparticles have also been explored for insulin. So let's put, I mean, maybe we are aware, but I would like to highlight insulin is the most widely explored protein compound so far for the different active transport technologies or delivery vehicles. The microparticles are very significantly explored for the delivery of proteins, peptides, as well as vaccines. So typically PLJ microparticles are reported for the delivery of cholera-toxin B. And as said before, regarding neural, it's very interesting, which contains cyclosporine. It has molecular weight of 1,200. As I said before, it is used as an immunosuppressant, and it's quite popular drug use during the organ transplantation as an immunosuppressant. There are two brands available for cyclosporine in the market. One is Sandimmune and Neural. And notably, both are lipid-based formulation. One is using the mixture of corn oil and ethanol. In another formulation, you see the presence of chromophore-EL and ethanol. But if you see the oral bioavailability, the neural shows better bioavailability compared to the Sandimmune. And that's the reason by using the new oral formulation, the observed bioavailability is significantly higher compared to the new Sandimmune formulation, because the problem with Sandimmune, it really depends on the patient population. So even in liver transplant patients, the observed bioavailability was less than 10%, but for the renal transplant patient, it was 89%. And the problem with immunosuppression, you really want to have the pharmacological response in the to achieve the desired immunosuppression. You also don't want the immunosuppression is so high. Maybe we have some issues in terms of the sensitivity of the individual towards the different infections. So this is also something to be kept in mind. The use of mucoadhesive polymers to achieve the delivery of this biologics. So in this case, what's happening basically, we discussed before the impact of the presence of the mucosa and how it really hinders the passage of the drugs from the intestinal medium to the intestinal cell walls. So in that case, the advantage is taken from the presence of the mucosa. So the drugs are encapsulated in contact with these mucoadhesive polymers. They are binding to the mucosal layer. These coated particles then passing through the intestinal cell wall, and maybe they have the possibility to open the tight junctions. Overall residence time is quite high. The mucosal layer is completely being replaced, and that's one of the advantage. If the residence time is quite high, we do have the possibility to achieve the desired delivery through the intestinal cell wall. So in that case, the literature reports different hydrogels are reported where it's possible to use the highly absorptive material, and they respond as a specific function of the pH changes in the GI tract, maybe as a function of the temperature, light, electrical changes. These polymers become responsive, and this effect is taken into account to release the release of the protein molecules from the formulation system. So these are listed as a stimuli responsive hydrogels. So whichever factor we want to use to trigger the release, this has to be carefully considered, and depending on that factor, then we have choice to select which typical pH sensitive or maybe light sensitive polymers, or maybe polymers which are responsive to electrical charge. We can think of using those polymers for the delivery. Then the site-specific delivery to the colon. The advantages of this approach: the colon has very less enzymatic activity. It works quite well for the permeation enhancer as a response. The transit time is quite high. The resulting pH is quite high in colon compared to the small intestinal, compared to the stomach. And that's the reason majority of proteolytic enzymes are not working quite well, or maybe the activity is quite less, and that's the major advantage. So in that case, typical strategy could be, we can use a typical pH sensitive polymers like eudragids. They mainly get solubilized or disintegrate at pH 6.5 to 7, which is a colon pH. So if we are trying to coat the proteins with these polymers, it's possible to achieve the colon specific delivery, so that maximum protein absorption is possible. But we also need to keep into account different factors or hurdles there, that the surface area available for the delivery into the colon is very limited due to the proximity of tight junctions. So the disease state of the patient could alter the activity of microflora we have there, and that could hinder the release of the molecules from the formulation systems. Product approach is something very important. I think we try to do it often for small molecules. We try to have a linker for a drug molecule, so that the linker is covalently linked to the drug molecule. It is passing across the cells, and once it is into the systemic circulation, the enzymatic degradation takes place, so that the drug molecule is relieved. It is more or less the same mechanism even for the proteins, and overall aim is here to improve its stability, improve its permeability, so that the maximum absorption takes place. But in this case, the typical challenges we need to consider: the proteins are very complex structures, and the reliable and reproducible methods are missing to obtain the products of protein molecules. Maybe in case of peptide, it's possible. These are like short molecular weight compounds, small molecular weight compounds, maybe 8 to 10 amino acids. Maybe there is possibility, we have possibility to use typical linker to one of the amino acids. But when you think of proteins or antibodies, this approach may or may not work. So it still remains a complex issue to be discussed. Then we can think of using cell penetrating peptides. So in this case, what's happening, we are just trying to bind to, may not be covalent bonding, maybe it's just formulation consideration. We try to achieve non-covalent bonding or conjugation of the cell penetrating peptides to the biologics we want to deliver. It reaches to the intestinal cell, and what happens, the cell penetrating peptides, they have the capacity to bind to the tight junctions, because these tight junction proteins are they do have some receptors. It's possible then cell penetrating peptides are binding to these tight junctions, and once the tight junctions are opening, then it's possible that the molecule which is attached to the cell penetrating peptide is passing across this cell walls, and that's how we try to achieve the systemic circulation. Interesting to note that even during the passage, this cell penetrating peptide is still bound to the biologics, and across the passage, maybe it's possible to trigger the release of the drug from the cell penetrating peptide. The typical examples reported into the literature are like penetratin or oleoarginine. These are quite widely used cell penetrating peptides for research purpose. And for the compounds like calcitonin or insulin, there are reports that significant increase in the absorption was possible when they tried to use the cell penetrating peptides. On this slide, I tried to list the different strategies which are explored so far for the oral insulin delivery. So I would like to highlight one thing. The first report for the oral insulin was published in year 1920. Today we are in 2020, and even in 100 years, it is not possible to deliver the insulin across the oral route. So you can imagine what we are discussing today, what challenges we are facing. But having said that, I mean, the research is still moving in a good shape, in a good direction, but there's still a lot to be done. And that's one reason also in these 100 years, a lot of literature already published for insulin using different technologies, different options. So there we can think of targeting neonatal FC receptor, transferrin receptors. So if the receptors are targeted, maybe we have possibility to improve the insulin delivery. The penetratin octagen are the cell-penetrating peptides I already discussed in the earlier slide. So if we try to use these peptides, we have the possibility to increase the oral absorption of insulin. Then you do have the possibility to use the active delivery technology. Basically, these are medical devices listed as SOMA and LUMI. I will discuss in detail in the subsequent slides. By using these medical device technologies or active transport technologies, it's possible to deliver the insulin across the oral route or across the intestinal or maybe stomach. Another option, maybe we can think of using the microenvironment responsive nanoparticles or the hydrogens, as we discussed before, using mucoadhesive polymers, which are responding to different changes with respect to pH, temperature, or electrical charge. So this is also something which can be explored. And on this slide, you see the different technology platforms already into the clinic for the oral delivery of the proteins. Most of these technologies have also used insulin. Another compound which is significantly explored is calcitonin, uh, ocosheparin, or some GLP-1 agonists. But then these platform technologies already come with a lot of information as they are reaching clinic, and the companies which are developing these platform technologies, they continue to explore this technology for other compounds they do have in the pipeline. So a lot of research is going on in this field. Sorry. On this slide, I tried to summarize the different technologies which are moving from, let's say, clinical phase up to the market. So you can see in the phase one, you do have some nanoparticles. And again, it's very important where you want to deliver the protein. Sometimes you just need to local delivery into the stomach, maybe intestinal. Often we need to deliver the drug into systemic circulation. And that also drives which typical technology we are using for the oral delivery of proteins and peptides. So for the systemic delivery, naturally, we are thinking to use penetration enhancers, the chemical modification, or maybe microemulsion based systems, or maybe the cell penetrating peptides. And when you are talking about local delivery, maybe we can think of using the protein secreting cells. We are trying to change the structure of protein molecule. Maybe entry coating is another option we can think. So a lot of research is going on. A lot of platform technologies are reported, and this gives one option for the drug development company, for the researchers to see which is the site we are going to deliver, what kind of protein we are dealing, and then that really helps to select the given technology for the further modification or further use. And on this slide, you see the commercially available oral peptides. So as we discussed before, the peptides, they are small molecular compound compared to the proteins or vaccines. So naturally, you have more and more compounds onto the market through the oral route. In this case also, I tried to segregate at least the compounds. These are systemically absorbed, the compounds which are locally delivered. So typically maybe vancomycin or some antibiotic type compounds or some drugs like colistin sulfate, you really want to have local delivery. We don't want to have any systemic delivery. For some of the peptides like triptorelin, maybe desmopressin acetate, you have the possibility to use these technologies so that the systemic delivery is possible. But at least it gives idea, or it gives a concept if you want to deliver a oral formulation for a given compound. And on this slide, we see the different biologics currently into the clinical trial, typically in phase 3. Maybe some they already are into the market, and some they are still into phase 3. And depending on the outcome, maybe we can expect they will be reaching the market very soon. So let's discuss the delivery options by using the active transport technology. And typically we are discussing here the medical device type of systems, or maybe the substance which have the capability to achieve the desired response at specific site, but we certainly trying to use specific energy form to deliver the drugs across the intestinal wall or maybe across the stomach. And the first example is of technology SOMA, self-orienting millimeter scale applicator. This technology was developed at MIT in the lab of Professor Bob Langer. So when they tried to deliver the technology, they really wanted to have some flexible shape. So they tried to keep in mind when they were trying to deliver this P-shaped device, which is called SOMA. The typical shape that they tried to consider is of this African tortoise, leopard tortoise, which tried to achieve the desired shape when it is tapped. And this is something they tried to achieve by computer simulation. So the device they have manufactured is mix of the polymers and still. So in terms of mechanism, how it works, so when it is reaching to the stomach, it tries to localize itself when its wall is facing the stomach wall. And as it is in contact with stomach, the fluid entrapment is happening. And so basically the needle is held in contact with the compress ring. There they also have the lyophilized sugar. So once the fluid is entrapment is happening, the sugar gets dissolved, the compress ring is relaxing, and that pushes this needle across the stomach cell wall. This needle is coated with the insulin, and once the needle is passing to the stomach cell walls, insulin release is happening, and that's how they try to achieve the desired insulin release across the stomach cell. The specific, the specific Madhur, can you mute all? Maybe it will be helpful. So what we are trying to discuss here. So the on this slide you see the needle shaft they are using. It's a 7 millimeter in length. It's a diameter in the range of 1 to 1.5 mm. They tried to perform all these studies in pigs. So this particular graph shows that the mucosal penetration is happening to this level in terms of millimeter, and the force they are using. This is a micro CT image of the needle. Here they have used barium sulfate as a contrasting agent. Even the needle shaft they are using, it's quite broad, maybe what they are using in practice, just to get a better overview. They also tried to do some immunohistochemistry, tried to see by using some dyeing agents, the dyes, that how this penetration of the needle is happening. And here you can clearly see from this histochemistry to what level of the stomach cell wall this penetration is happening. On the right side of the slide, you can see some PK studies they did compared to the subcutaneous injection of the insulin compared to the intragastric administration of this drug against soma. And then you had like a control group, no injection. And you can clearly see the soma technology was behaving as good compared to the subcutaneous injection in terms of the plasma concentration of human insulin. In terms of percent glucose change, also you can see the soma was as good as compared to the subcutaneous injection. So the maximum dose, I think they have explored to insulin is 3 milligram, which they were able to deliver. And in fact, this is also the therapeutic amount the patients are using through the subcutaneous injections. So the technology, I think, very well accepted even by FDA, and they are all set to start their clinical trials. So very fantastic technology. Another example I tried to put is of LUMI, so the luminal unfolding microneedle injector. So in that case, what they are doing basically, they came up with the capsule dosage form. So the capsule contains this microneedle type of patch systems, and which has a capacity to propel these microneedles. So the microneedles are coated with insulin. So in terms of the mechanism, what's happening? So maybe we can go to the next slide. So when the this capsule is reaching to the small intestine, sorry, the water enters the bottom system, and it leads to the opening of the capsule shells. The microneedles are propelled, and once the microneedles are propelled, they are traversing across the intestinal cell wall. The needles are already coated with the insulin, then the insulin release is happening, and that's how they are trying to achieve the insulin release across the intestinal cell wall. And these are some images from their ex vivo studies, how these microneedles are located, how this device is located when it is reaching to the intestine. This is the typical image of the device. Here you see the insulin coated microneedles. Important to highlight, once the drug is released, the non-biodegradable part of the device is excreted. In terms of the PK profile, you can see they tried to compare the intestinal microneedle patch against LUMI. They also tried to use the gavage of this dissolved microneedle, and the subcutaneous injection was also used. So the LUMI system was behaving as good as the subcutaneous injection of the insulin. So we can say that the device was able to deliver the insulin in the therapeutic amounts. And then the question comes, what's happening in the field of biosimilars, biobetters, because at the end of the day, we also need to think what are the costs we are dealing in terms of the sourcing of the typical API. So when we think of discussing the any innovative drug, the cost we are dealing in the range of more or less 1 billion US dollars. Maybe for biosimilars, it's not the cost, because maybe we are using in that case different cell lines. Maybe the exact sequence may not be the same, but at least at the end of the day, the the desired physiological response we want to achieve in the same range. We don't want to have any immunogenicity. So the time frame and the cost for the biosimilars is not same. But when we are thinking of biobetters or biosuperiors, the investment is also quite high, because there we are trying to come up with the maybe better potency, enhanced shelf life, or also improved safety and immunogenicity. And that's the reason that the significant efforts are needed. And at the end of the day, maybe you want to come again with the superior compound, maybe significant investment is needed there in order to come with the highly potent, highly stable compound. And naturally then it is going to have impact on the research we need in terms of delivery for any new compound. At least in the biologic segment, they try to come up with the IV or parenteral formulation, and then the next option is whether oral formulation is possible in that case. Then to let's discuss few slides about vaccine before I try to conclude my presentation. So oral vaccine delivery is also very important aspect we should consider. So there we need to deliver the immune response to the oral route. So the microcapillaries across the local and peripheral cell layers is very important there, and this is something we like to target to achieve the desired response. Another option, maybe we can target the lymphatic drainage to have this immune response, or the M cells from the pairs patch is another target site we could achieve. Naturally, this is not possible for all the vaccines. It really depends on the typical structure of the vaccines and typical structure of the protein molecule. But at least you still have some vaccines onto the market today, which are still given by the oral route, like poliovirus, rotavirus, as well as typhoid and cholera. So depending on the structure, depending on the target site, it's still possible to achieve the oral delivery of the vaccine. The particulate systems already we discussed before, so I just would like to quickly summarize that the use of liposomes, polymeric nanoparticles, or emulsion is possible. The typical advantage here that the antigens are coated, so they are quite stable. If you want to achieve the combination of the antigens with adjuvants, so this is also one thing is possible. And paracellular transport is certainly possible, or maybe this tight junction catalyzed active transport is also possible in this case. Typical example of the oral delivered vaccine is a typhoid vaccine. So it's very interesting to note that by oral route, the typhoid infection is happening. It's quite easy. So the researchers tried to use more or less similar mechanism to deliver these vaccines across the oral route. They simply tried to change the virulence of the antigen, but the rest of the delivery mechanism is still same. And in this case, they are trying to target the M cells in the pair patch. So what are the challenges we are talking for the clinical translation of the oral biologics? So we need to really carefully control the design and performance. How robust is the formulation or the technology, its safety and scalability, and what is the cost benefit ratio we are talking at the end of the day. Significant safety data is needed. What a device or technology we are using, what are the regulators expectation there we should try to fulfill. At the end of the day, we also need to fill the cost and timelines expectations from the management, and this is something to be carefully considered. As I discussed in the beginning of presentation, finally you would like to have the solid dosage forms where we may go for freeze drying. So how it impacts the protein structures as well as the cost and timelines, this is to be considered. And a lot of information exchange is necessary between the innovator companies where they are trying to develop the compounds, because the protocols are not so easy to design. So there should be open communication so that it's possible to compare the different technologies by using the common platform. And then with that information, I would like to conclude that although there are few clinical use technologies which are existing today for the oral delivery of these proteins, peptides, vaccines, but despite of long research activities, if you see the scale of challenges, it is still very high, and there is still significant need to overcome the barriers we are talking for the oral delivery of the biologics. The chemical absorption enhanced strategies is very limited to, and maybe we do have very in vitro success there, in vivo we still need to prove there. The nanotechnology option still shows very interesting potential for the delivery of oral biologics, but there are still safety issues, delivery capacity issues, regulatory expectations are quite high in terms of reproducibility, the, let's say, the accumulation of nanoparticles, what's happening, the in vivo effect, for example. And then the future growth in the research activity of oral biologics, especially with respect to the delivery devices, is really it will be facilitated by use of electronics, robotics, and materials. We briefly discussed before this technology, LUMI and SMA. So maybe this will set some benchmark to come up with the new systems, new technologies for the delivery of oral biologics. So with this, I would like to finish my talk. Thank you for your attention.
Thanks a lot, Dr. Yogesh, for a very, very informative and a very interesting topic. It is really challenging. And not only, I mean, the oral delivery of biologics is not only challenging, but talking on it is equally challenging, I believe. You indicated the huge market that is available for this, for generally for the biologics, and particularly for the oral biologics. You went on to explain the physiological barriers that are there for the absorption of the biologics, then the formulation considerations, importance of the stability of the biologics, and the formulation development challenges as well. Then you also very well explained the variety of the approaches that are there or that have been tried for enhancing the absorption, oral absorption of the biologics, such as penetration enhancers or particulate drug delivery systems, mucoadhesive delivery systems, colon targeting systems, pro-drug approaches. And then you went on to explain very, very new technologies like SOMA and LUMI, which would probably see the daylight of the market very soon. The case study of semaglutide that you explained was very, very interesting, and it's very nice to know that this particular biologic is actually approved by US FDA for oral delivery. And yeah, you also explained briefly about the oral delivery of the vaccines and the possibility of that. So it was quite an extensive extensive presentation, but you tried to cover each and every aspect of it. So I would like to really thank you for such a nice presentation. And if you're okay, we can take up a few questions now.
Yeah,
Because I
please.
can see a few questions in the chat box.
Yeah. So would you take questions, or shall I answer on my, I mean, how we should proceed regarding questions?
Oh yeah, if you can just read it up, you can just answer, no worries. So the first question is from Dr. Pranav Shah. So do you see any possibility of oral insulin getting translated from bench to bedside?
Yeah, so I think this typical example we saw of Soma technology. In that case, at least they were able to deliver 3 milligram insulin by using the specific technologies, which is also they claim to be a therapeutic dose. Very soon they are to start about their phase 1 trial. So I mean, if things could go well with respect to safety and efficacy, maybe then we have chance to also deliver insulin by oral route.
Yeah, okay, thank you. Professor Vishakha Hastak would like to know more about the microneedle approach for the drug oral delivery of biologics. Of course, you have explained the microneedle approach, but the thing is, the question, I mean, the added question to that is, are these microneedles going to actually pierce across the cell membranes of the GI lining?
Yes, yes, yes.
But how can one ensure that penetration?
I mean, if you see the typical this LUMI case study I have showed, maybe I can also share the screen and we can go to the slide. Give me 1 second. Can you see my screen here?
Yeah, yeah.
Okay. So at least in this case, they were able to show by using this micro city techniques that when so they are not basically coating this needles in that case with insulin. Maybe they are coating with barium sulfate, the radio
Yeah.
contrasting agent. And this is typical graph or image you see that these microneedles are reaching across the depth of the intestinal cell wall. And that's a clear localization that microneedles are piercing across the cell walls. And that's how it is possible to deliver the any therapeutic active you are coating on the surface of microneedle.
Okay. Yeah.
This is example microneedle, and in this case, okay, it's not microneedles. They are using some needle shaft for the Soma technology, but here you see this is the stomach cell wall. This is the
Okay.
device they are using where the spring is actuating the needle movement, but you can clearly see how this needle is piercing or it's penetrating across the stomach cell wall, and how they are able to achieve the delivery of the therapeutic moiety across the cell wall.
Okay. So they've been able to show this or prove
Right,
it that
right.
it does pierce. Okay.
And that's only able to drive the PK response you get, right? If it
Yeah.
is not
Yeah.
possible to open this cell walls or the cell membranes, naturally there will be no absorption. If there's no absorption, we will not see any PK-PD response.
Yeah. Right. True. Then there's a question from just a moment. There's a question from Dr. Rahul, NMIMS, Hyderabad. He's saying that you have mentioned about chitosan being an option for hydrophobization, but its toxicity is a major challenge. That's what he's saying. And then also FDA approval will be a problem if you use chitosan.
Right.
So are there any other acceptable alternatives?
Right. So at least we discussed that there's a lot of research happening. We are for a lot of options. We are still not into the clinic. Once we are in clinic, then the FDA discussion is starting really. So from research background, I tried to show what is possible, what are alternative options. If you are, it's not possible to use some of these polymers due to safety concerns. Other possibilities I tried to highlight. You may try to change the backbone proteins. You may try to change the backbone protein into the form of more lipophilic properties rather than the hydrophilic properties. That gives you one chance to have the hydrophobic characters. Other possibility to have some linkers, so you are going to have the covalent bonding with the typical lipidic components. You have the possibility to use the lipids such as capric acid, the lauric acid. If you try to use the linking of your protein molecules with these lipidic components, it's also possible to achieve this hydrophobization.
Okay, thanks. Thanks for that. And one more question from Dr. Rahul. CPP is important tool applied in the delivery of biologics, but it suffers from endosomal trapping. So is there any route to streamline this problem?
So in case of endosome, the use of CPPs, if you see the mechanism, so it's typically trying to activate the tight junction transport, and that's how it is possible to achieve the absorption of this oral biologics. So, and as you highlighted correctly, that finally the absorption mechanism is through endocytosis, and that's how the molecules are traverse across the intestinal cell wall. So there is a problem of endosomal trapping, which may be also linked to the, because endocytosis is a physiological mechanism. So naturally, maybe there are no possibilities to change that physiological mechanism, but depending on the CPP typical peptide you are using, maybe you have the possibility to look for alternative options, maybe with different peptides. It is possible you are not into the similar problem you highlighted here with respect to the endosomal trapping.
Yeah, okay, thanks for that. And one from my side, you did mention about colon targeting, and it at times is a better option as compared to targeting to intestine because of lack of enzymes. I mean, there are not many digestive enzymes present in the colon. But what about the enzymes that are secreted by the gut flora?
Right. So the enzymes secreted by microflora are still there, and this is also to be kept in mind when you are trying to deliver the biologics at that specific site. But what we discussed that the nature of the enzymes or the concentration of the enzymes is typically low. What we are trying to discuss compared to the stomach
Right,
and small intestine,
right,
and most important, your transit time is quite high there.
right.
So
True.
enzyme, at the end of the day, enzyme is also a chemical substance. So once the enzyme is consumed, then you do have sufficient material at the site of action, and due to the longer residence time, you still have some possibility to deliver the drugs at that particular site.
Yeah. Okay. Yeah. Thank you. So I think that's all. I mean, I don't see any more questions in the chat box, and I would once again like to thank you on behalf of the executive committee as well as on the behalf of all the attendants for such a fantastic presentation. So we have come to the end of this technical session. And I would now request all the non-members, I mean, those who are not the active members of CRS-IC to leave the meeting, to leave this meeting now. That's because very soon we'll be commencing the AGM of CRS-IC.
Thank you all.
Yeah. And Mr. Ajit Singh, I mean, sir was there throughout the session actually. So if sir is here, then I would I would like to request Mr. Ajit Singh to please address the members of CRS-IC.
Madhur, I think he's left.
Oh, he's left?
He's left. Yeah.
Okay.
He's left. So
Okay.
we can carry on. You can carry on with the next session.
Yeah. Okay. So then we'll, in a minute or two, we will just proceed with the AGM. Yeah. Pete sir, you can just stop live.